So we're going to do something really exciting here. We're going to talk about drug delivery in 3 different sections. We'll start with glaucoma, and then we'll have 2 retina sections. And first, I'm going to ask our panelists in the glaucoma section to introduce themselves. Barb, why don't you start?
Thank you, Kuldev. So I want to say, Park City's my home. So it's so great to see everybody here. I know I actually practice here. Like, how lucky am I? So Barbara Wirostko, I'm an adjunct professor at the University of Utah. I'm a glaucoma specialist. I have an appointment in biomedical engineering. I was at Pfizer overseeing the Xalabrands portfolio. And I'm also co-founder and chief medical officer of Qlaris. And we're developing an NCE to lower IOP as a topical.
Anthony?
I'm Anthony Khawaja. I'm a glaucoma specialist doing the medical and surgical care of adults at Moorfields Eye Hospital in London. I'm also a professor of ophthalmology at UCL, and my research group does a lot of genetic epidemiology, which is driving discovery of new targets, and we're working with collaborators on hopefully developing new molecules, but also using the genetics to predict and subdivide glaucoma patients to see who will respond particularly well to some treatments and not others.
Randy Craven, I'm a glaucoma specialist and was in clinical practice for a number of years and worked with a lot of people over the years through clinical trials and developing companies. Actually, Rick Hill was a resident of mine, and I watched him start Glaukos, and it went from there. And had a chance to move off to the Middle East for a while, where I was the chief of glaucoma in Saudi Arabia at the Kekesh Hospital, and then went back to Johns Hopkins, where I was an associate professor in charge of multiple practices throughout the Washington, D.C. area. And then during COVID, decided it was time for a change, and Allergan and AbbVie were just in the process of merging, had a chance or an opportunity to come in to be involved with the company. And now I'm a senior medical director for research and development broadly, but that goes anywhere from medical affairs to clinical development or discovery or business development as well. So all that.
Hey guys, Cody McKenzie, Vice President of Global Marketing Strategy and Franchise Management at Glaukos, responsible for the glaucoma, corneal health, and pipeline commercial business. I've been with the organization for about 8 years. Prior to, I was actually at Allergan. I was the Director of Marketing for the Zen glaucoma implant. Prior to that, I spent a fair amount of time at Bausch & Lomb, where I managed the intraocular lens and viscoelastic portfolio.
Well, thanks. Thanks, everybody. So this morning, Kristen showed us the ophthalmology business pie chart. $50 billion business. Glaucoma pharma is about $5 billion of that business. Devices and everything else, about $1 billion. So even in this very heavily genericized world, pharma still dominates in terms of revenue. But drug discovery is here. Drug delivery is here, I should say. And Randy, why don't you lead us off on telling us about the Durysta journey, because you guys were first to market in this space.
Yeah, thank you. Yeah, you're right. You know, if we look at what our leading growth product is on the RX side, it's Durysta, which is drug delivery. And if you think about it, all of us know the challenges with patients for treating glaucoma. And when we first started off looking at how to put something in a place that would be successful for controlling IOP, you know, we looked at subconjunctival, long-term topical delivery forms, all sorts of things. But really the thing that makes most sense is drug to target. That means somehow getting the drug intraocular. And when you start off with the basics of, well, how long does it take for a drug to elute from a platform of some sort? And then you have to redose based on that. That's kind of where we started off with Durysta. I think we had to redose so frequently with it. And then now that it's gone on for a period of time and hit approval, over 4 years ago now, that, we had a lot of real world experience. And what we're finding is that the concepts that we kind of went into the studies with, which were designed that way based on a typical period of time where we thought it would dissolve, what we found is that it's actually the effects have gone on much longer, and through our real world evidence, we're finding that people are getting out to 18 months plus of IOP control with it. And that's been, really good as we looked at that from our Argos study that was just published a few months ago. So, so it's evolved and it's still evolving, and we're continuing to look at new platforms and methodologies to help deliver drugs safely and effectively to the eye, which gets into all other concepts of what's the best drugs to use inside of the eye for drug delivery.
So Cody, clearly this is still an area where there's growth opportunity. Extended release therapeutics and glaucoma is still a very small business. You guys have now launched iDose. I believe it's sort of a slow, controlled rollout. Can you give us an update on the journey and tell us how you're doing?
Yeah, sure. So actually a lot of similarities of what Randy was just talking about, but iDose itself, the idea came up in 2008. So fast forward, it was a 15-year journey from an ideation to FDA approval. And as everybody in this room knows, once you get that FDA approval, the real journey begins from there. So we're about a little over 14 months into the launch now. We did take a very controlled strategic approach to the marketplace. But I think we perhaps underestimated a little bit, as I think many new novel therapies do, the path to coverage and payment, which dictates a lot of commercial appetite and integration in practices. So we've been very focused on establishing coverage and payment for the product. The clinical demand is there. I think the desire for procedural pharmaceuticals and drug delivery is absolutely there, whether it's Durysta, iDose TR, or future therapies. But we're in the throes of that right now. We're very happy with how the launch is going. It's a synergistic, I think there's an opportunity for all products to exist, Durysta, iDose, and future, because it's not a one-size-fits-all approach. But our goal with iDose was to deliver a long-lasting therapy that can provide 24/7 benefit to the patient in a safe and efficacious way. And I think we're seeing that play out very well in the marketplace right now.
So Barb, Cody's talking about unmet need. You practice. I mean, I don't know how you do it. Be a CMO of a company and run a busy glaucoma practice. What is the unmet need here? And why is this not bigger yet?
Which is not bigger yet?
Why is this market not bigger yet? We've had Durysta out there. We've got iDose. Tell me what the unmet need is and how you see these products.
So I think something that Cody said is really spot on is with all the opportunities that we have now, whether they're surgical innovations, angle-based surgery, topicals, or sustained release, we are now treating patients on a personalized level. So we're really trying to understand the level of their disease, their unmet needs. And I've used Durysta. I've had patients that have had iDose at the VA. Actually, we started to offer it Salt Lake City at the VA. It's a tough sell. 3 years is long, and I'm not saying that there's not value there, but I think for the average patient, I've had patients say to me, but I'm doing well on my prostaglandins. They're not that expensive. So why would I want to undergo a procedure? So I think it's finding the right patients. I've had patients with Parkinson's disease, multiple medications. If they're going to be going on surgery soon, Durysta is perfect to kind of cover that time period. And I think there's still an unmet need. I'm going to put in a cheer for EVP and Qlaris because it's that personal approach. We're on a prostaglandin, but you're only getting IOP to a certain place. We've got a MIGS that's an angle base, a Hydrus, but we can still go lower. So there's that distal resistance that I think still is an unmet need for our patients.
So Anthony, you don't have extended release therapeutics yet in Europe, UK, certainly not in the UK, but I don't think anywhere in Europe. But you do have a big trial on laser trabeculoplasty, the LIGHT trial, which is kind of changing how people are practicing in the UK. Laser was shown to be at least as good and maybe even a little better than eye drops first. So now you have drops where there's clearly challenges with compliance and side effects. You've got laser, and now you have extended release therapeutics. How do you see this all playing out in Europe?
So maybe it's worth reflecting on how well lasers rolled out in Europe, and I think our experience in the UK also mirrors it across Europe. I think a lot of people are suggesting it's the best first line. We personalize our approaches. Clearly, for some patients, if the starting IOP isn't that high or their angle's quite narrow, you may start with drops. But for most people, we're starting with, or at least offering, SLT. Interestingly, not all patients want that. Some patients, when given the option, would still prefer drops. Then with SLT, you've got to remember a quarter to a third won't respond, so they'll need to start drops. And then out of the ones that do respond, in the real world data, looking at the real world data, maybe half have failed by around 2 years. So certainly we're getting used to delivering glaucoma care without drops, or starting off at least without drops, but actually nothing lasts forever in glaucoma care, and people often need to go back on drops. So I think medications are here to stay. I think adherence remains a problem. In Europe, where drops are cheap and really very easily available, I think it remains to be seen whether an intraocular delivery of a drug that can be given so effectively, topically and safely, how well that will work. And unfortunately, we won't find out for some time, I don't think.
So Randy, eye drops therapy has been a very profitable business for AbbVie. Certainly it was for Allergan and for AbbVie. What I find curious is that in the last 28 years, there's only been one new therapeutic class of medications introduced. So you, as a practicing glaucoma doctor who's seen the evolution of medicines, now seen extended release therapeutics being introduced, where's your investment going to be at AbbVie? Are you willing to look at new novel entities, new molecular entities as eye drops, or is it only drug delivery now going forward?
Our discovery team has looked at a lot of other potential molecules for being able to be available. The problem is, though, is how good that the prior generations of drugs have actually worked. And the bigger problem on top of that, though, really the bottom line, if you look at what the industry standard cost is to get a drug to market now, from, all the way from early discovery through approval, industry average is approaching over $2 billion, okay? So, if you have a generic market where the pricing sets the model for things and you're trying to compete against that, it's trying to balance that out, how you can ever try to get something like that to work. And it's not easy. So yeah, there are targets that have been identified, but trying to get that safety profile and efficacy profile enough to get the value high enough to bring it to where it needs to be is not easy. That's the real challenge.
Cody, I was going to turn it back to you on iStent and how iStent fits with iDose. Clearly, there's an opportunity with cataract surgery when you're implanting implantable devices in Schlemm's canal to also deliver a drug. Do you see this as being a paradigm that you expect the company to see grow? And in fact, how do you think things will shape up over the next 5 to 10 years for you guys?
Yeah, it's a great question, one we talk about quite frequently. And, as we've talked about here, there is no one size fits all, right? So whether that's starting with an SLT or a procedural pharmaceutical or a MIGS, or even, for a more advanced patient, microinvasive bleb surgery or a filtering surgery, that's on the table. But as we think about stents versus procedural pharmaceuticals, we would argue procedural pharmaceuticals probably make sense as an earlier in the paradigm treatment option because of what Randy was talking about. These are good drugs, they're proven pharmacologics that work really well, and they provide great efficacy while providing a good balance of safety, and you're giving that patient 24/7 delivery of a proven pharmaceutical that has very high responder rates. Now, whether that's done as a standalone or in conjunction with cataract, the sheer number of glaucoma patients that are out there, it would be a shame if we just continue to stay in doing it with cataract surgery. There is such a standalone opportunity out there that remains significantly underpenetrated. So as we look towards the treatment algorithm, starting with a procedural pharmaceutical, then moving on to a MIGS like an iStent Infinite, which is approved for standalone utilization, that's kind of how we see things playing out in the future. Or doing them in conjunction together, frankly.
Thanks, Cody. Yeah, so question related to molecules. So prostaglandins are once a day. There are other drugs that are twice a day, 3 times a day. There are other drugs that we can't even use on the eye because the side effects are terrible. Is a prostaglandin the best class to make a once-a-day drug, or should we be looking at other molecules and saying, hey, let's put this in the eye because then we don't have to dose it 3 times a day? Barb, you want to comment on that first?
Sure. That's a really interesting question, right? And I think time is going to tell, right? We just don't know if a prostaglandin at a high dose in real world is really, truly safe for all our patients. And I think, Cody, because I've been thinking about this, I have one patient who has iStent, Hydrus, goniotomy, pseudophakic, Zen, and finally got a PreserFlow. I mean, I have never seen so much hardware in one single eye. And you've got to wonder, when is too much, right? So again, the cataract with the prostaglandin, are we going to predispose those patients to CME sooner if we're doing cataract surgery and putting in a high level of a prostaglandin, which will cause inflammation in some patients. Time is going to tell. I am very conservative by nature, open to new ideas, but conservative.
Anthony, you want to comment on that?
Yeah, so I mean, I think that's a really good point, that if one of the main advantages of putting the drug inside the eye is convenience, then going for a drug which you only use once a day, maybe you should go for something which is less convenient. And especially because you've got to balance it against having to undergo that procedure and the risks, even though they're very small. So it's something that's either less convenient or something that, as you say, Kuldev, you can't give topically. So I'd be quite excited in the future about new modes of action or things which have a bigger impact on convenience.
Randy,
Quick
you wanna
comment on that I'd like to.
Yeah, please, yeah, go ahead.
You know, the target that we're trying to achieve with these different things and the concentration of drug that's required. If you take a look at, trying to work on, a beta blocker, for instance, the drug load that you need is quite high because it's a continual need for that. With the PGA, we were able to put in basically inside of the Durista implant the same amount of drug that's in one drop of the 0.03 Lumigan, roughly, very close to the 10 microgram amount. So if you're able to decrease drug, which can help avoid side effects and other things, that's great. But when we start looking outside of the PGA pathway, it gets very hard with the amount of drug that's required to figure out how to deliver that. So you get a continued action, and the payload, if you will, is so big that it's hard to do that.
Yeah, and just to add on to that, this is publicly disclosed, but we've been working on trying to get a ROCK inhibitor in the iDose canister for probably 6 years, right? It's not easy to do these things, to your point.
So great point. So we got about 1 minute, quick lightning round. And so Barb, we've got a drug on the market or an implantable on the market that's good for 6 months. Can't repeat it yet. We've got an iDose up to 3 years. What's the optimal amount of time for a drug delivery therapeutic in glaucoma? Because we're going to be seeing these patients also, and when you go longer, there's a trade-off. So
I would say a year. I like a year.
Anthony?
I think it depends on the stage of glaucoma. We can't afford to see our low-risk patients more than once a year in the UK. And so if you're going to have to bring patients back more often than that, then you're going to have to limit it to the patients you were going to see more often. That's a smaller market. So I would say at least a year as well.
Randy, your thoughts?
Different competing thoughts. So one, if I was a patient, I'd want a once and done, so I'm out of there. But looking at the realities of caring for glaucoma patients, I think somewhere in that frequency of intervals where it falls within visits is probably acceptable.
Cody, last word?
Yeah, I'll quote our CEO here. When we were developing iDose, it was, if we could get 6 months out of it, that's fantastic. If you get anything more than a year, what a slam dunk. So,
Great. Well, this was kind of a lightning panel, and I'm going to ask Bob and Lejla. Thank you to the panelists. Bob and Lejla are going to come up and next, Retina in the office.
Thanks, Kuldev. I'd like to invite Lejla and my co-moderator and the panel up. And while they're working their way here, I'd just like to make a comment as a retina doctor. I don't think anything has changed the scene of ophthalmology, at least during my career, maybe before my career when the IOL came out. But nothing's changed it more than in-office administration of anti-VEGF drugs. I mean, I used to see a lot of disciform scars when I started practice, and we just don't see that anymore. And it's because of this advent of giving drugs in the office. With that has come a horribly complex treatment burden that we have practicing. And we have here a who's who of experts from companies that are trying to minimize that for us. We have 2 companies making TKIs, which are inhibitors of VEGF, slightly different from the antibodies. We have 2 gene therapy companies that are making a biofactory producing aflibercept to again reduce the treatment burden. We have a company that's mastering how to deliver macromolecules in an intravitreal injection. And we have the premier company with optogenetics, which is a slightly different way of administering things, something to create a new sort of photoreceptor in the cell that has the ability to improve vision potentially in a whole host of inherited diseases, and that therapy is gene agnostic. So welcome, panelists. I'd like you to introduce yourself. Why don't you go ahead and start, Sam? Why don't you start? Go ahead.
Thanks, Bob. I'm Sam Barone. I am the Chief Medical Officer of Nanoscope Therapeutics, doing the optogenetics therapy that Bob just talked about.
Hi, everybody. This is Ramiro Ribeiro. I'm the Chief Medical Officer at EyePoint. At EyePoint, we're developing a potent TKI, vorolanib, in Durasert technology, and now we're doing a phase 3 study, Lugano Lucia.
Hi, I'm Peter Soparker. I'm the Chief Operating Officer of Adverum, and we are one of the two panelists up here. I am one of the two panelists up here that is doing the biofactory and the eye approach gene therapy.
Hi, I'm Michael O'Rourke, CEO of Re-Vana Therapeutics. I've been in the ophthalmic drug delivery space for 30 years now, originally with Chiron Vision. We launched the world's first ever sustained release intraocular device, Vitrasert, back in '95, and then with Bausch & Lomb, we launched Retisert in 2005. So, good to meet you.
Good afternoon, Dhaval Desai. I'm the Chief Development Officer at 4DMT, developing gene therapy expressing aflibercept as well as a VEGF-C silencing mRNA.
And I'm Nadia Wahid. I'm Chief Medical Officer at Ocular Therapeutics, and we are developing extended release potent TKI for retinal vascular disease and wet AMD.
Thank you. Lejla, why don't you start?
Yeah, fantastic. I'm Lejla Vajzovic. I'm retina specialist at Duke, and I'm really honored to co-moderate this panel. Really, we have a powerhouse of leaders really in the ophthalmic space, but also working very actively on longer duration products for us, really more options for our patients. Nadia, I would love to start with you and kind of addressing, I know you have really been passionate about developing longer duration products for our patients with really this sustained release platform. What in your opinion, where do the TKIs essentially fit in terms of retina specialist practice? Where do you see them fitting in? And how do your registration trials potentially help us understand how this product will fit in and what needs would it address?
Absolutely. So, I think that extended release for treatment for wet AMD has been the holy grail for a very long time. We know that the current mechanisms of action we have work really well, but don't last enough, and that that leads to suboptimal results for our patients. And so at Ocular Therapeutics, of course, we're developing AXPAXLI, which is basically a TKI in a hydrogel, a TKI inhibitor in a hydrogel that provides extended release 6 to 12 months. And I think one of the things that I'm really excited about is the innovation that we've done in clinical trial design, really, as we develop this product. I think the group at Ocular Therapeutics, as you know, you require 2 clinical trials, 2 registrational trials to file with the FDA to get approval. And the general fallback has been that people design 2 trials that are identical to each other. And of course, so when we started with our SOL-1 clinical trial, the expectation was that we would have SOL-1 and then we would have SOL-2, which would be an identical clinical trial. And we decided to take a slightly different pathway. So we went with these really bespoke complementary clinical trial designs where the 2 clinical trials are different from each other. And the idea there was that to create synergies that help answer all of the different questions that patients and physicians and that really payers might have around the mechanism of action and the efficacy of the medication. So if you look at the way the clinical trials are designed, we can answer questions around durability, and that's the SOL-1 study, which is once a year, right? We can answer questions about a comparison to standard of care, which is the SOLAR study, non-inferiority study compared to standard of care Eylea every 8 weeks. We also can answer questions around comparability to Eylea high dose. We have the ability to answer questions about whether it's safe to redose. The SOLAR study, as you know, goes on for 2 years, redosing these patients. So we believe that we really kind of have leveraged the synergies of having 2 trials. Now, the other interesting piece is that this enables us to have possibly one of the best labels out there, right? So a label that gives us a superiority label first and foremost, which is the first of its kind in this field. It also enables us to have very flexible dosing between 6 and 12 months between the SOL-1 and the SOLAR study. enables us to redose our patients after the 6 to 12 month label, right? That and the fact that you go to the FDA with this very diverse data set, and whenever they look at the studies, remember we have to present both registrational studies together, when they look at the result of the non-inferiority study, they're doing that in the context of also seeing the SOL-1 study, which was a superiority study, and knowing that this medication works and has a large durability of action. So we really believe that other companies will follow this really innovative approach of doing complementary trial designs rather than doing identical photocopy trial designs.
Fantastic. Thank you, Nadia.
Ramiro, I'd like to ask you to follow up with your TKI. You probably had a slightly different opinion in the trial development with respect to the draft FDA guidance about shams, but I'm really more interested in what you see if these drugs, these TKIs get approved. What do you see the clinical utilization being versus the label? I mean, we retina doctors are notorious for off-label use of everything. Do you think this will be used as the trials are set up, or do you perceive it being used in a different fashion?
Yeah, no, thanks for the question, Bob. And I think one of the important things with our program and the development of TKI is that we're bringing a new mechanism of action for patients with wet AMD. So it's an inhibition of intracellular VEGF receptors, and with that, trying to bring additional benefit for patients. We've seen in our preclinical study some data showing neuroprotection, delayed development of atrophy, that we're also going to be looking for in our phase 3 program. Now, going back to your question in terms of utilization, our approach is that our phase 3 studies are designed to show that patients have the same visual outcomes as standard of care while reducing a treatment burden. So I think physicians are going to continue to use the same standard of care that they have now. Most physicians use treat and extend. So with DURAVYU, they're going to follow the same path, right? So they're going to bring the patient, they're going to first treat with anti-VEGF, then they're going to give DURAVYU, they're going to bring the patient back, and then try to extend that as much as possible. Our study is designed to dose every 6 months, but our data have shown in our phase 2 study that 50% of the patients went all the way to 1 year without getting any anti-VEGF.
Great, thanks. Michael, you've been in drug delivery for 30 years, using small molecules from ganciclovir up to TKIs. What made you tackle the hardest thing, the giving macromolecules, which has been so difficult? I mean, I remember ITAC promising microspheres 20 years ago and for Macugen, and it seems to have been an insurmountable mountain to climb. What's prompted you to do it, and tell us how you're succeeding with Re-Vana.
Yeah, thanks, Bob. So clearly with the introduction of the anti-VEGF products from 2006 onwards, it became clear that that was really going to be the mainstay of retina treatment. So became aware pretty soon that the use of frequent injections in the eye was a major burden, both to physicians and to patients. So I joined Re-Vana in 2017, really with the primary goal of taking on board a totally untapped market opportunity, and that was really to deliver a 6-month sustained release biologic in the clinic without surgery. So that was the idea back then. We knew there's major barriers to overcome when you're trying to develop sustained release biologics. One is you have to ensure that there's no degradation of the protein, so there needs to be good protein stability. You need to have sufficient release rates to obviously release enough drug to treat the condition. You need high drug loading, a high percentage of drug loading by weight, so you get enough drug into the implant itself. You need to, in our case, you needed to really think about degradation, which has to be controlled and accurate. And lastly, you have to think about tolerability, because the single biggest reason for any drug delivery technology failing is because of inflammation, because of poor tolerability in the eye. So we have managed to overcome these 5 main barriers, which has caused a lot of failures in the past. And the way we've really done it is we have taken on board a photocross-linked, biodegradable hydrogel implant. So we use light instead of heat, and that helps protect the protein itself. So we're able to do this with 2 technologies. One is called iLeaf, which is preformed before it goes into the eye with ultraviolet light. It can deliver 2 micrograms or less. Our second technology is one called Oculeaf, which is an in situ forming gel. When you put the gel into an aqueous environment like the vitreous, it forms an implant. So we've developed a proprietary applicator that allows us to both deliver drug and to photocross-link it in situ, but this time with visible light instead of ultraviolet light. So that's the basis of what we have really taken on board to deliver sustained-release biologics in the office.
Fantastic, thank you. I'm going to switch gears towards gene therapy and longer duration. It's exciting to see gene therapy entering as an in-office procedure as well. Peter, I'm going to ask you for a really quick question is like, where do you feel Adverum program fits in terms of what patient population, entry patient population, would you say is best fitting for this program?
Yeah, thank you for the question. And I just want to take a little bit, take a moment to take half a step back. And Michael, my fellow panelist here, is talking about anti-VEGF in 2006, right? That's longer than we've had the iPhone, right? That's 20 years where improvements and what we've been talking about is 6 weeks versus 7 weeks or 8 weeks. And suddenly up here on this panel, we have 3 different options that 3 years from now, the conversation is going to be radically different in what we're able to offer patients. We don't all always see eye to eye up here, pun intended, in the approach that we want to take, but we all are after the Holy Grail, which is bettering options for patients. So coming back to the question that you asked, of course, you know, the stock answer for gene therapy is everybody, right? All patients with wet AMD. There are some realities and some facts underneath that. You know, one, we do anticipate, and I think as with all new modalities, you expect that your first patients being treated are the ones that are the highest need, your frequent flyers. That's always going to be true anytime you have a new modality. And I think there's another reality too. When you have treatment-naive patients, you don't know what their treatment burden is going to be. You have patients that come in with wet AMD and they get 1 or 2 injections. They don't need any more. Is that the right patient for a gene therapy? Maybe not. So I think that, you know, really on the one hand, I say yes. Every patient with wet AMD, it's going to be an obvious answer. But I think the reality is probably patients that need a few injections a year, because ultimately we all know, everybody in this room who treats patients or is engaged with patients or talks with patients with wet AMD, they hate injections, right? Their greatest desire is to be injection free. We know that. We talk to them. I mean, I can look out and I see all of the treating physicians nodding their heads. They hate it. And that's ultimately what we're all trying to get to. We're trying to get to these patients being injection-free. Some of us, I think, are on a step towards that. I personally believe that gene therapy is the answer to, and really the only answer right now that we have to being injection-free. But that's ultimately the goal.
Thanks. Dhaval, you're also in the gene therapy quest for wet macular degeneration. What does your company's drug for DMT, how do you distinguish yourself from the other companies? And maybe tell me how that helped you select your phase 3 trial parameters.
Yeah, it's a great question, Bob. And I think the first thing that sets it apart is how we approach building the compound, right? And so using directed evolution, the capsid that actually houses 4150, R100, is completely different from anything else we've seen before. And we didn't kind of pretend to know by putting this mutation in or that mutation in that it was going to be able to do the things that we wanted it to do. We created a library of billions of different capsids and we pushed it through a biologic system. And at each step, we removed the capsids that weren't doing what we wanted it to do. And mainly, there's 3 things that you're looking for, right? You're looking to minimize the immune response, you're looking to selectively transduce cells of the retina, and you're looking to be able to do that via a simple intravitreal injection. And I think that those 3 things have really manifested itself when you look at the clinical data, the low rate of inflammation, when you look at the non-human primate model, predominantly retinal cells that are transduced, and you see sustained vision and control of anatomy over a period of 52 weeks, with a lot of these patients being injection-free. And to what Peter said, you know, it's the only option that we have up here that ultimately gets patients to where they want to be, not getting injections.
Fantastic, thank you. I'm going to switch gears to optogenetics. Sam, I'm going to put you on spot here, but asking how does optogenetics traditionally differ from traditional gene therapy delivery's intravitreal approach? What are some of the similarities or differences between the two?
Thanks for the question. So gene therapy is essentially taking genetic material and then getting that inside the cells for the cell's own machinery to then release a protein. And so with, first think about the protein that's being expressed by that genetic material, by that gene therapy. So when you look at my colleagues up here, their protein is an exogenous protein, like we use in optogenetics, means it's not a protein that's naturally made by the cells. In contrast to traditional gene therapy, where you're looking at replacing the cells, replacing the mutant gene, and that's releasing a deficient protein. So that's sort of the first thing. The similarities and differences you're looking at. What's the protein? Then where do you want it expressed? And that's based on the function of that protein. So again, at Verum and 4DMT, they have a secreted protein that is going to impact the retina and to the choroid to be able to decrease the choroidal neovascularization and impact the permeability of the vasculature, et cetera. Other gene therapies, traditional gene therapies, they want those to be into the specific cell. When I think about Luxturna, for example, we want that expressed in the cell where it's going to have its impact. Now, with optogenetics, our protein, it's a membrane-bound opsin protein. And so we want that to be expressed in our target cell of interest, which is the bipolar cells. So the inner retina cells are intact in advanced retinal degenerations. The photoreceptors are lost, and that's how you lose vision, but the intact cells still remain in the inner retina. So that's our target cell, and that's where we're looking to that's where we want to express. So then I think the final thing, all three of us here who are in gene therapy, we are all intravitreal delivery, and there's a certain advantages to that. There can also be disadvantages, or there can be challenges, and again, that's based on your target cell. And so that's why there's other gene therapies that utilize a subretinal delivery and a surgical approach. All these are some of the nuances and the different considerations in when you think about gene therapy in the eye. It's not just gene therapy, everything's the same. There's a lot of different considerations that we think about.
Thanks, Sam. I have a generic question for the panel. We're running out of time, so just the first one or two people to give a short answer. Inflammation has been a problem with a lot of drugs and anti-VEGF world, and some have died because of it. We've been spoiled with how safe they are in general. Any of these extended therapies may come up against this and would be deleterious for adoption. What is your company doing to try to minimize that? And anyone can answer this.
I'll go first, because gene therapy, we see it, right? And we know it's a potential issue. And so what we've done is, I think most gene therapy programs are doing some twist on this thing, but Durezol given over a 20-week taper seems to be able to control that inflammation very well. Obviously, the important thing in any phase 3 program is to monitor those patients very, very closely.
Great.
And so
anyone else? Because we're down to 30 seconds.
Yeah, I would say that, I think drug delivery, as I said, is killed by inflammation. So we've done multiple animal studies to really show that tolerability is mild to moderate in all cases.
Yeah, we've dosed lots of humans and not seen any evidence of inflammation so far.
Great. And Peter?
I do think it's going to come down ultimately, as with all therapies, to benefit risk.
Okay.
If your benefit is in 6 to 10 years.
I have to let Sam.
Yeah, I was going to say, the benefit risk, and for example, we happen to be in the rare disease space, right? So there's a real different benefit risk calculation compared to looking at a mass indication like AMD.
Great. Well, I want to thank the panelists and Lejla, my partner here, and invite the next group up. It'll be Alan Ho and Namrata Siraj. We'll be talking about delivering these drugs in the operating room instead of in the office. Thank you.
There are some seats scattered over here in case any of the people that want to maybe prefer to stand. That's fine. So this particular, as I think about my career and the transformational things that have happened, modern day cataract surgery anti-VEGF in the office, OCT imaging. Those would be the three that I would talk about. And we've made great strides in the office with injectables in the retina space, but some of the unmet needs may require pharmacosurgical delivery. And so we have a great panel here, and my co-moderator, Dr. Namrata Saroj, who has deep industry experience and knows all of retina very well. So let me have the panelists, starting with you, Mark Humayun, introduce yourselves.
Okay. Mark Humayun, Professor of Ophthalmology, University of Southern California.
Ron Polanki, Chief Commercial Officer at REGENXBIO.
Dante Paramici, Retina Specialist, California Retina Consultants.
Hi, Rich Small, CEO of Neurotech Pharmaceuticals.
And I'm Brian Cully, the CEO of Lineage Cell Therapeutics.
Thank you all for being here. Now, we're going to start off with Rich.
Thank you.
You have a new approval, and as it turns out, as I think about this, this is a cell, genetically modified, and a surgical implantation all in one. So you really kind of cover all of the aspects of this panel in some way in your approval of Encelto for MacTel. But it hasn't been an easy journey. It's been a couple decades. So maybe you can tell us a little bit about it and give us some lessons learned.
I have 17 minutes to talk to you about this. Yeah, I know. As you said, Alan, you know, we, for the folks who don't know, we got approved early March for Encelto to treat the orphan disease, macular telangiectasia. You know, it is a long journey, lessons learned. I'm not even sure where to begin, but you know, I will tell you that the whole FDA process, you really need to pay attention and work closely with the agency. They're very excited about new technology. That was our experience. And they actually gave us a pathway to actually approval, which was very important. As Alan said, drug device eluting a therapeutic, and also understand this is a one-time treatment. So, you know, this implant is in the eye potentially for several years, decades. And so a lot of focus on the safety aspects of which the other panel talked about, and all was very much attention, and you know, we had very good answers and worked very collaboratively with the FDA on that.
When is it going to be available?
It's going to be available in June. And just a quick plug, we have a commercial team that we've been forming over the last couple of years. We actually, I'm just coming from our commercial launch meeting, and product will be available, as I said, in June, and so we're working with all the constituents to get as seamless of a launch as possible.
I'm going to switch to Dante from a physician's perspective. You've been extensively involved with the PDS system, and now you have all these other long-term delivery systems, and some of them in office coming in. How do you see the PDS system, the advantages and disadvantages with the new delivery methods?
Yeah, I've been involved with port delivery system, I guess, since the initiation of the clinical trials some years ago. And it's like every other product, it's gone through a big evolution. I think it's become safer. I think that this has now been indicated for DME, diabetic retinopathy, hopefully soon, and neovascular AMD. And it's a device, I think, that can consistently deliver drug every 6 to 12 months for a patient, once or twice a year, within office refills, but it's a surgical procedure. And that's been a hurdle, I think. I mean, it's been a hurdle for the uptake of the device because it takes training. There's a lot of logistics with getting the device in the operating room, which Rich is going to have to deal with as well. So these things are hurdles that are much easily solved in the office with an intravitreal injection that we're used to. I think the landscape since the PDS program begun has changed. I mean, we now have things like gene therapy, which I'm very excited about as well, which may be every 6 months or one and done type treatment. We have TKIs that are, and again, these are going to be office delivered for the most part, also potentially in the operating room, but for the most part, office delivery. So again, you have a patient in the chair, you have A or you have B where you have to go to the operating room. With that said, I think the port delivery system really is a platform therapy, and it can deliver all kinds of medications going forward. And there may be medications for which this finds a home, and people will breach those hurdles, go to the operating room, because that'll be the best option.
There's been a lot spoken about the desire to meet the unmet need of the burden of treatment. And yet, we have patients that come in frequently, every 4 weeks, every 6 weeks, every 8 weeks. I think what the patients really want is, yes, less burden, but better vision still. And monotherapy, repetitive monotherapy, anti-VEGF therapy is tough to beat. It's tough to beat. Ram, are there any signals, and this is for Dante as well, that the different pharmacokinetics of gene therapy, a lower steady state dose of anti-VEGF therapy, either with PDS, Susvimo, or with gene therapy, could yield better vision in years 2, 3, and 4, 5, and beyond, where we see those vision curves just plummet?
Yeah, I think we published this data, right, Alan, and you've been involved with this since the beginning. Some of your patients that have gone all the way out to 4 years and 5 years now. When you look at our phase 1/2 data, and I think what we're observing is even when you take patients that were previously treated and have gone through a process of treatment burden and frequency, for some reason, when they actually switch to gene therapy and you look at their long-term outcomes, that consistent expression of a therapeutic protein and how you manage the disease might have a disease-modifying effect. I mean, we're seeing in patients that have had disease for 5, 7 years before, now coming into the next 5 years post-gene therapy, vision gains that are about 1 to 2 lines on average. So I think that trend seems to be very supportive of, to your point, it's just not about decreasing the number of injections, it's about providing clinical value on top of it.
You see that as well, Dante?
Yeah, and I think there is some data talking about disease modification from the port delivery system, that perhaps the progression of geographic atrophy may be slowed down when you have consistent low-level suppression instead of these wide swings up and down. So I do think that this continual low-level suppression may be disease modifying and may reduce the progression of things like geographic atrophy in the AMD patients.
So Ram, I just want to build on that, and you've witnessed, you've been part of the drug development for over 2 decades, and you've seen sort of the adoption of clinical trial endpoints to clinic. Where do you think we stand now, and what are the more important endpoints that will actually dictate clinical practice?
I mean, vision comes down to be the basic primary, right? But how you look at vision, I think, is going to be important. I mean, this is the first time we have multiple clinical trials that are designed in a different way and looking at different endpoints, even from a vision standpoint, right? We started our life with, looking at proportion of patients that lose vision to proportion of patients that gain vision. In the middle, the mean change in vision, and they're all coming back now. I mean, it's even on the basis of what you guys are trying to do, I think being creative about how do you bring the value forward. It's going to be very interesting in the next 2 to 3 years. When you look at all these clinical trials, the different endpoints, the different phenotype of patients that are enrolled, and then the outcomes you're looking for, I think clinicians will have a lot more burden to really understand these things and think about how they apply to them, to their patients in the clinic. So great evolution. I think this is also going to create more innovation from a standpoint of how you approach regulatory development, and you know this, superiority trials. Every superiority trial has failed in retina to date. Exactly.
Fingers crossed.
Yes. But I think the superiority approach also has to be different, and this is the first time you guys are exploring that, and that might become a path for a lot of the people as we move into the future.
Stem cells in the lay press, generating a lot of hope, maybe unfounded hope. Stem cell centers across the country, charging patients, lack of data, and we've lost eyes with that. And I think we're in a place where responsible stem cell research is being done to meet unmet needs like in geographic atrophy. There are a couple approaches up here on the stage. One is with cell suspension, and another is with a sheet. Brian, do you want to tell us a little bit about the evolution of your particular program at Lineage?
Yeah, so as you mentioned, we employ a suspension of RPE cells to disrupt how we treat dry AMD patients.
Surgical.
Yes, thank you. What we have seen, and we're really excited that others have now independently shown this in early studies, is that an RPE transplant can generate clinical outcomes far beyond the reach of anti-complement therapy. So not just thinking about slowing the progression of the disease, but actually in many ways reversing it. And that includes bringing back functional activity. And I agree. I think 20 years ago there was a lot of optimism, but it was perhaps tremendously premature. But now we're seeing, as I say, multiple independent validation of this approach. But the delivery is a very important component. One of the things that Lineage noted was that delivery of the cells right onto the area of atrophy was associated with the best outcomes. And so our partners Roche and Genentech are working with proprietary devices to really figure out what's the best risk-benefit profile for delivering these cells, whether transvitreal, there's next generation orbit device, really exciting approaches, which I'm particularly excited about because it's not asking a question about efficacy. It's asking a question about how do you optimize a surgical procedure, what tools, methods, et cetera. I think the risk of that is very different than asking whether an intervention has activity.
When I see these geographic atrophy patients with loss of vision, and I tell them, and we're involved in many of the clinical trials, I say, you have a couple approved injectable medicines every couple months. It is not going to make your vision better. It is not going to stop vision loss. It's going to slow the growth of this age spot that I'm showing you here on the monitor, and maybe we'll have some vision benefit over time. But the seduction of some of the signals that you've seen, I think you've seen as well in sheet transplantation. So why don't you tell us a little bit about where you are and maybe contrast the approach.
Well, yeah, I mean, I think if it's going to just slow down the progression of geographic atrophy, then obviously there's no room for cell-based therapy because we have anti-complement. So I think you're exactly right, Alan. I think if we're going to go to the operating room and really put cell-based therapies in the OR inpatient sides, it's got to have a significant upside. And as Brian just mentioned, the upside is really regaining vision. But how is that happening? It's very interesting. Our work over the last 7 years has shown, and that's why we're using a sheet, is from work in AMD going back all the way to Marco Zarbin's, Bruch's membrane is affected. It's just not the RPE. And also the choroid capillaris is affected. And obviously the outer segments are affected. So it's a really a big area that we're dealing with. These cells are not just working at one level, they have to really work at other levels. So the data we showed and why polarize and why go through a substrate to implant this bigger procedure, more complicated, is that polarized RPE cells, if you take that same exact cell and it's not polarized, it creates 2% of the pigment epithelial-derived factor, PEDF, or VEGF basally. So it just doesn't produce the growth factors that are necessary when they're not polarized. Then in terms of their survivability, they don't survive as much or as well when they're not polarized. They incite an inflammatory reaction when they're not polarized. So, nobody wants to make something harder than it needs to be, but it was very clear to us that you need to make it polarized, you need to have a monolayer there, and that led to the work we've done. And our phase 1/2A has shown that 27% of the subjects, it was a small study, as you know, 15 subjects, improved in vision, and out of those, it was a mean BCVA gain of 8.2 letters. And not only did they gain that at year 1, but we just published it 3 years out, they maintain the vision. So those cells don't, and that's the other thing. People always worry, you're putting these cells in a very hostile environment. Are they gonna all die just like the host cells did? But they don't, and there's a lot of studies about that too, how embryonic stem cell or iPS-derived RPE are more robust when you put them through various stresses.
Are they?
They are. We did peroxide testing to adult RPE cells and to stem cell-derived RPE cells, and they're much more robust in terms of how they're able to survive. So polarized cells have a distinct advantage. Stem cell-derived RPE have a distinct advantage, but clearly it's a surgical procedure, and we have to make that as safe as possible.
I have a question for Rich. So the bridge between drug development and physicians using it is good commercialization. We're heading into this era of niche markets. We just talked about, maybe advanced GA here. You are launching in a niche market. Maybe you can comment on what steps are you taking to be successful in that?
Yeah, so you're absolutely right, where we have are developing just a very comprehensive approach where just if you think about it, we have to deal with the physicians, their admin, patients, the payers, which obviously is a whole big issue. And then because it is a surgical procedure, we have to deal with the ASCs. So we have put together a very comprehensive portal system where our goal and intention is to once, basically as best we can handhold both the patient and even the physician through the whole process and to demonstrate and make it as seamless as possible. I think that's, and on top of that, this is not something we're just going to blast out. We're actually starting with a very stage-controlled approach. We need to get this right. We think we have something that can be incredibly important to the community, and we just want to make sure that we launch it properly, done right, and then it will become accepted. So we're very attuned to that, and we just need to make sure, I'm sure it's not going to be perfect, and we'll adapt as we see what the results are, but we are it's right on the top of our minds right now to make sure we do this properly.
The details of some of these efforts will be in the breakout rooms, which is our next session in 1 minute and 20. Any last comments from our speakers? Dante?
Yeah, no, I'm excited about all these things. I think as a clinician, it's nice to have options, and I think the options are looking better and better. As has been said 1 million times, anti-VEGF therapy delivered intravitreally works great in the neovascular AMD patients. I think that with Rich's device, it's very exciting. I give him a lot of credit. I've heard about neurotech for the last 25, 30 years, and he wins the Cal Ripken Award, the company does, for sticking to it. I think, for those who have young companies out there, stick to it because eventually it'll come.
Come talk to me. I'll go through all the nightmares and wars.
Okay, we're going to end 29 seconds early, and we're going to break. There are 3 rooms, and there they are. Anterior segment glaucoma, Deer Creek, retina gene therapy and novel mechanisms of action with Namrata and I and Strawberry, and TKI and drug delivery with Mark, Lejla, and Bob Avery and Jordan L. Thank you.